Alrighty, you all can take it away.
All right. Wasn't sure if that was everybody. Okay. Hi, everyone. Welcome to the ARO seminar series centered around middle ear. Today, we're delighted to have our last speaker of this topic, Dr. Pita Santamaria. My name is Dr. Jen Chisholm. I'm a research audiologist working at the NIDCD at the National Institutes of Health, and I'll be the moderator for today's seminar. A little background before we get started about Dr. Peter Santamaria. He is a clinician scientist in otology and neurotology with a focus on translational medicine and commercializing medtech and biopharma. Dr. Santamaria earned his PhD in molecular biology from the University of Western Australia in 2012, specializing in wound healing of the tympanic membrane in the middle ear. His research on identifying a novel treatment for chronic eardrum perforations was accelerated through the SPARK program at Stanford University, which is a unique partnership between university and industry experts, winning the Excellence in Stanford SPARK 2014 award. Subsequently, he partnered with Aration Biotech and Astellas Pharmaceuticals to advance the novel treatment, bringing the non-surgical option for repairing eardrums into human clinical trials by 2020. He currently serves as the director for SPARK Global and helps SPARK programs in over 30 institutions worldwide, with a focus that includes translating innovations in low-resource settings. In addition, he is one of the founders and chief of the Scientific Advisory Board for Aration Biotech, as well as the founder and chief medical officer for Flowtherm. He also serves as the division chief of otology and neurotology, vice chair of translational and clinical research, co-leads the UPMC CREATES Lab, and is the emeritus chair of the American Academy of Otolaryngology's Medical Device and Drugs Committee. So a few announcements before we get started. If you have any questions during the presentation, please type them into the chat and I'll address them during the Q&A session. Following the presentation, feel free to just unmute your mics and directly ask your questions. And any questions that can't be addressed during today's session will be answered by the presenter in a follow-up email. Closed captioning for this meeting is available, and the session is being recorded and will be made available for additional viewing in the link in the chat. So without further ado, it is my pleasure to welcome Dr. Pita Santamaria, who will be presenting his work titled Changing the Paradigm for Therapy in Chronic Suppurative Otitis Media. Over to you.
Thank you very much, Jennifer. Thank you. I always get embarrassed when I hear your introduction. So thank you. I think that means that I have no personal life. So thanks to everyone for welcoming me. And I also, it's super exciting. I just saw there's a lot of participants joining there and a lot of friends and colleagues that I really love talking to. And, you know, anytime that you want to hear me talk about what I'm passionate about, you're more than welcome to grab me aside. So, you know, I'm really excited. I've just joined the team here at Pittsburgh. For those of you familiar with Pittsburgh, it's an incredibly exciting team, and it's growing, and we're hiring. So please reach out to me and Thanos and the other researchers here. If you didn't know me already, if you didn't tell by my accent, I started here in Perth, Australia. And I'm loving the weather here in Pittsburgh at the moment, but it's hard to compare to the weather in Perth, Australia. And when I see these pictures, I forget how beautiful it was. And people ask me why I ever left. But this is where I kind of cut my teeth on otitis media. So, you know, I'm a clinician scientist, and we would do these trips to regional Western Australia. And you kind of follow the arrows there. We would fly from Perth right up to, you know, the middle of near Halls Creek. And you kind of see we would change these desert regions, you know, to Australia where the climate would really change. But, you know, just to give an idea of size, that's like flying from San Diego to Montana for an ear clinic. So, you know, the size of Australia is really huge, and the broad coverages, and you know, it really affects the delivery of healthcare in the region when you have to cover so much area with the resources that you have. And you know, just like you saw these beautiful pictures of Perth, this is the same state. You really change the climate in the country that you live in. And this is where a lot of the Indigenous kids live, up in that area. And you can see they live in very different social conditions. And this is where they had the highest rates of otitis media in the world, with a 45% incidence of chronic otitis media. And we would do these ear clinics with the kids. We would see something like 100 patients in a morning. And almost every child had one of these three appearances. And for those of you in otitis media would recognize this as otitis media with effusion, tympanic membrane perforation or inactive chronic suppurative otitis media, and chronic suppurative otitis media down the bottom right. And this is a spectrum of disease, and this is something that we're all aware of. And even though that we concentrate on different parts of otitis media, it really needs to be understood as a spectrum of disease. And so what I'm talking about today is mainly concentrating on a lot of the work that our lab has been doing most recently in chronic suppurative otitis media. And so as I alluded to, chronic suppurative otitis media is a huge burden around the world, and it is predominantly affecting the peri-equatorial region of the world, including the bottom of Central America, the top of Australia, Asia, Africa, and things like that. And I think this is another benefit of being a clinician scientist, is that you can do the clinical research as well as the translational basic or discovery research. And it's also a lesson for a lot of the scientists out there. We're working in clinical departments or with clinicians, and one of the best advice that I was given is that sometimes you just need to collect your own research and your own statistics. So this really helped with the significance portion of the grants that we write. So we went and did some epidemiological studies, and it is limited, but we're able to somewhat quantify what the burden is in the United States. And not surprisingly, it does match other first world areas. Its prevalence is about 0.46%. It does have some regional differences, being slightly worse in some of the border states and the warmer climates. But if we go back to Australia, the standard of care for a chronic suppurative otitis media is what we call dry mopping, which is really sticking a Kleenex in your ear, sucking out the pus, and putting in betadine in there. Interestingly, one of the big initiatives that was done in the communities was putting chlorinated pools in the communities, and it not only improved attendance at school, but it gave the children's ears a wash every now and again, which actually reduced the burden of otitis media in the region as well. So, very basic standards of care out in the rural communities. But how do we currently manage it, in the University of Pittsburgh or in modern clinics? Is we often see these ears that kind of look like the top left. That's what you're looking at, is a tympanic membrane. It's full of pus, and you can see the inflammation there in the middle ear, and it's pouring out. We often take a swab of that. I actually use a lot of betadine still in the clinic today. That's another topic for another discussion. But we use acidifying agents. We use white vinegar, boric acid, other powders, and things like that. And there is only one approved antibiotic, and it's the fluoroquinolone. Afloxacin or ciprofloxacin is a fluoroquinolone that we often use to treat in the ear. So we have limited treatment options in the ear. And so what I'm hoping to do with this presentation is kind of change everyone's understanding of why these treatments fail and what we could potentially do about it. So chronic ears are just chronic bacterial infections. They really represent, when you look at the bacteria and the bacteriology, they represent a lot of other areas. So chronic lung disease and cystic fibrosis, chronic rhinosinusitis, diabetic wounds, these are all chronic bacterial conditions. And a lot of bacterial conditions that we have might lead to sepsis, might make the host very sick, but these kind of live almost in there, because I guess if they became more invasive or worse, they would lead to the host dying, which is not really in the bacteria's best interest. But it's still a problem, and they still cause a lot of functional disabilities and a lot of potentially mortality down the line. But really, why don't these chronic infections get better, and what could be potentially do about it? So the first problem is that when you measure bacteria from the middle ear, we typically take a swab of the pus and we send it off to the microbiology lab, and we send it for culture. And this is typically what we get: no growth. But we still typically treat it with antibiotics. So I'm just going to take a little segue, is that a lot of what I'm presenting to you is a collaboration from a number of universities from around the world, in the US, from Egypt, from Iran and India. And we've collected a clinical isolate bank of over 150 clinical samples of CSOM. So a lot of what I'm going to talk to you about and what we've learned is from around the world. So the first thing is that it's often polymicrobial. So if you look at that, there's also regional distributions. So in Perth, where I used to practice, it was almost exclusively Pseudomonas. In Stanford, it's a mixture between Pseudomonas and Staphylococcus and other gram-positives. In India, there seems to be a flip between some of the more bad gram-negatives like Proteus, Enterococcus, and E. coli. Egypt seems to be a very high gram-positive region. Interestingly, when you look at the epidemiology of this, it seems to be very regional, and even neighboring countries in Africa can have very different bacteriologies. But I think the thing to point out there, the two that rise to the top most commonly are Pseudomonas and Staphylococcus aureus. The other thing to consider here is that this is not swab results. This is amplicon sequencing of RNA through PCR. And so what we're detecting by culture is not what's representative in the middle ear. So when you take a swab, you really only get what's grown in laboratory conditions in that particular microbiology lab. There may be other species there, and there might be other things there that we don't know about. And really only by PCR are we really detecting the burden of disease. But as clinicians in the clinical setting, unfortunately, we're not doing that. So let's just change our clinical setting. So let's pretend now that instead of having a negative result, we had a positive result in that culture, and it's showing what we typically might grow as Pseudomonas. In fact, in this patient, we've grown 3 strains of Pseudomonas. And we look, what the clinician typically does is they look across to the susceptibility template, and they go, well, look, hey, we got Pseudomonas there. Yeah, ciprofloxacin, I recognize that medication. It looks like we've got this susceptibility. The MIC is 0.5 micrograms per mil. You know, it says susceptible. We should be able to treat it with ciprofloxacin. So the first thing is to understand what that actually means. MIC means minimum inhibitory concentration. So when you grow the bacteria on a plate, they have increasing concentrations of that drug exposed to the bacteria. And the lowest threshold at which it stops growing is determined to be the MIC. It's not eradicating the bug, it's inhibiting the growth. So that's MIC. The other thing that is determined is when they give you the sensitivity cutoffs, which is around typically between 4 to 8 for fluoroquinolones. It's based on the serum or the maximum serum concentration that can be achieved, which really is not relevant for topical consideration here. So when you look at the bottle label drug of 0.3%, which is about 3,000 milligrams per mil, it is way above the MIC reported concentration cutoffs for ciprofloxacin. So if you just think about this as an MIC story and a bacteria story, why doesn't the fluoroquinolone kill the bacteria when the MICs are way above what is needed? And if you look at the clinical literature, it's very, very poor. And they're all short-term results. In fact, if you look at a Cochrane review of this, they say that if you look at the inclusion criteria was at least 1 week follow-up. And so all of us that deal with chronic otitis media know that 1 week follow-up is not sufficient for a chronic condition. So it's obviously showing that there is a lack of studies that are really even looking at this. But the clinicians in the room and those of us that work in chronic otitis media know that these things typically fail to resolve, and there's even published rates of 70%. But we do know that fluoroquinolones work in some way, and that they're converting from active to inactive around 90%. And so if we look back at Stanford's results of how successful that was, and remember that Stanford usually is taking people who have already been treated by the community already, is that patients typically treated with medical management recur, at 50% of them recur. You can see that the dotted line 50% recur around 4 months. So they're not lasting long. We're not keeping them inactive for very long. And the burden is actually significant. On the left there, you can see that on average over a 5-year period with about almost 200 patients, there's about an average of 10 otology visits, several prescriptions, almost 2 surgeries at a cost of up to $20,000 per patient. So like I said, 3,000 micrograms per mil is way above 0.5 micrograms per mil. So why doesn't that work? So the first is to understand drug kinetics. How much of that bottle-labeled concentration actually gets into the middle ear and where it needs to be? So if you sample the middle ear mucosa, and micrograms per gram is equivalent to micrograms per mil in this setting, 3,000 actually dilutes way down to 400 micrograms per mil at about 8 hours. In fact, there's high patient-to-patient variability. If you look at the concentration inside the mucosa, and remember the mucosa is very inflamed and itself acts as a barrier to drug penetration, it is undetectable in many patients. But in some, it's still 250 micrograms per mil. So if you look at that story and you were to end here, you think it should still be effective in at least a portion of the patients, but that's unfortunately not where the story ends. So our lab developed a model that we could show this in and understand why this disease relapses. So we use chromosomally encoded Pseudomonas. We inoculate that using a special recipe into the middle ear, and it becomes chronic suppurative otitis media. We can then use an in vivo imaging system to measure them externally. You can see there in the middle picture there. And it sort of, we've false colored it red, so it just glows red on the IVUS, the in vivo imaging system. So we can tell if the disease is active or not. And just to tell you, this tells you whether the disease is active and dividing. It doesn't tell you whether the bacteria are there, because there is a lower level of threshold cutoff. So if we treat these guys with fluoroquinolones for 2 days, for 2 weeks, twice a day, just like we do in humans, you can see that the IVUS signal goes away. And this is exactly what we see in humans. We, inverted commas, cure the disease. And just like in humans, if we were to stop the treatment, it relapses. And so here in the mouse, our model relapses within 3 days. But the other thing to point out in the model, just like in the human, the lack of middle ear effusion does not mean lack of infection, the presence of bacteria. Just because you can't see it with your eyes doesn't mean it's not there. And another takeaway from chronic otitis media, chronic suppurative otitis media, is we need to assume the infection is there, even if you can't see it or is potentially active at that time. So the first thing is, should we believe the MICs? So here we have a table of Pseudomonas lab strain, Pseudomonas, one of the clinical isolates, Staphylococcus lab strain, Staphylococcus, one of the clinical isolates. And we have the MICs according to the common preparations that we use. We have a fluoroquinolone, an acidification agent, iodine and boric acid. And just to help orientate you, the green means it should be susceptible. So the MICs are all within range of what the bottle concentrations are. So we should be able to control these bacteria if we were to believe the MICs. And if we look at these strains, these are graphs of the CFUs and the reduction of CFUs after various exposure to ofloxacin. And you can see that there is over a 3-log reduction in CFU, which means that they're susceptible. So all these strains are susceptible to fluoroquinolone. So resistance is not the problem in this setting. But one of the first things to understand is as you start to get away from that bottle of concentration, you start to get dilution of exposure. And you can do this using a disc diffusion assay, for example. So if you were to load the bottle concentrations onto the disc in the middle, as it perfuses away from that agar, away from that disc in that agar plate, you get reducing concentrations. And what you end up with is a zone of inhibition. If you can see there at the top left for an example, you can see a little bit of a zone of inhibition of growth around that top disc, which means there is some susceptibility to that lab strain at reducing concentrations. But below it in the clinical isolate, you can see the bacteria growing right up and onto the plate, meaning that maybe it is susceptible at the very highest concentration, but it's not susceptible at the lower concentrations. And you can plate it like this. And this is one of the things that we're trying to change in clinical practice, is to kind of switch to this disc diffusion assay for assessing antimicrobial susceptibility. And this is another key slide that we're going to learn about with chronic otitis media. And this is how we currently understand bacteria. So when we have bacteria, just like cells, if we were to grow them, They sit there sleepy for a little while in their lag phase, and they grow in the exponential phase. And as they run out of their nutrients in the plate, they form the stationary phase, and you can graph that. This is the planktonic cycle of bacteria cells. And these are the infection that you see that are swimming around that cause the pus and the excessive infection. We test this measuring the MIC, the test that I just explained to you. We test this in plates, and this is the current understanding of antimicrobial bacteria. So antimicrobial therapies aimed at bacteria. Many of you would have heard this before as biofilms, and these are the bacteria defense to attack and how they establish chronicity. So they go from their planktonic form, from this their free floating form. They lose their flagella. They form these communities. Within these communities, up the top, they may be a little bit more active. Down the bottom, they can even change their metabolism to become anaerobic. And when it becomes sophisticated enough, they can even break off again and become planktonic. There you can see them swimming away. And they protect themselves between a slime matrix. We do not test biofilms in clinical medicine, and even though that we're dealing with biofilm-related diseases. So this is one big problem with clinical medicine and treating chronic otitis media, that we don't test for this. So how can we treat this? A new concept to many on the talk here is this idea, and if there's one takeaway from this talk, it's really understanding this third concept. It's that within these biofilms and within other cells, is that bacteria can change their metabolism. They can become metabolically inactive, and we call these cells persister cells. And these cells are tolerant, and they're tolerant to antibiotics because they're not metabolically active. And all currently available antibiotics act by metabolic mechanisms. And so, therefore, if the cell is dividing, it can act. But if they're not, and they're metabolically inert, then the site of action does not exist for that antibiotic. So that it's important to understand that persister cells are tolerant but not resistant. If you treat bacterial populations with antibiotics, it usually clears away the planktonic infection, the metabolically active cells, but it leaves behind the persister cells, and these persister cells repopulate with the same sensitivity as before. And just like biofilms, we also don't test this in clinical medicine. So at the core of chronic suppurative media is an understanding that persister cells are what make infections unresponsive to antimicrobials. So like I said, the antibiotics comes in, it doesn't have a target, it cleans away most of the infection, which is why you see the drop from active to inactive, and the persistent cells remain. When the antibiotic goes away, the persistent cells proliferate. But over time, the mutations can happen within these persistent cells. If you continually passage these persistent cells through by not killing them, but killing away the metabolic counterparts, sometimes they will develop resistance mechanisms, and so eventually you will end up with a resistant and a persistent infection, and the resistant cells do not even react to the antibiotic in the first place, and therefore you're not able to even control the active component of disease, which is where a lot of us who manage chronic suppurative otitis see is that over time the ear just stops responding and it stays wet. Interestingly, if you were to separate those persistent cells, and so how do you get persistent cells? Well, you determine the MIC of the bacterial culture. Usually treat way above the MIC, maybe 50 times above that, and then you regrow what's left. And you see that when you regrow what's left, you measure the MIC, and it should be unchanged from before. So you know that they're not resistant, but they're persistent. And when you regrow these up, they're a little bit more sleepy. You can see there's an extended lag phase here. They also generally, when you compare them to stationary and exponential phases of growth of their counterparts, they usually have lower ATP. So these are the only real defining the features of persistent cells. They're not phenotypically different, but they're metabolically different. And so this is just showing you that how we get persistent cells is that we treat them way above MICs, and what you're left with is a tiny, tiny fraction. But clinical isolates can evolve to be higher persistent cell producing bacteria. So a little cartoon just to explain what happens there is the antibiotic comes in, it has a binding site, it binds to the binding site, causes cell death. And this is typically how antibiotics work. Resistance happens when that binding site, or firstly happens when the bacteria develop or upregulates efflux pumps. So the binding site's still there, but it just spits out the antibiotic straight away. So you need to overcome that with more antibiotics. An alternative there is a modification in the binding site where it can't bind, and you need to overcome that with more drug. Persistence is where the binding site is not there because it's not metabolically active. Fluoroquinolones operate on acting on DNA gyrase, which happens during a cell division. So if the bacteria are not dividing, the target is not there. Because the target is absent, it has no effect. Sometimes it can cause damage to the cell still. There's still mechanisms for antibiotics to cause damage outside their primary mechanism of action. But because the cell is sleepy, it has time to repair. So there are two ways that persister cells get around that. So if you were to overcome resistance, you just pour more drug onto that. And that's how you see elevation of the MIC. Persistence, as I mentioned to you, and what a lot of approaches have to be, is they feed the persister cells, they wake them up, they show that the target's still there, and what happens is then you measure the MIC, and the MIC is actually unchanged afterwards. So the difference between resistance and persistence is measuring the change in the antibiotic binding, which is a surrogate for that is measuring MIC. So ENT surgeons typically look at this and we treat the patient, they get better, inverted commas, and they go away, but then they come back. And unfortunately, what you're doing is you're winning the battle, but you're losing the war. And I'm not going to talk about this, and this is a significant area of research in our lab funded by the NIDCD, is why we lose inner ear hearing here. We're all very familiar that we lose conductive hearing because we're affecting the ossicles and the tympanic membrane, but what is less appreciated is the sensory hearing loss for that. And this is just an example of a patient, and if we look at, unfortunately, she didn't want to have any treatment for her ear, but she did see me 5 years later, and we saw that her bone thresholds had dropped slightly. So it's not a dramatic loss, but it's slowly affecting over time. The other thing that can happen is that we can develop antibiotic resistance, and if we passage these over time, eventually the MIC goes up, and that's just showing you the story of increase of resistance over time. Interestingly, they don't just develop resistance to the parent antibiotics, ciprofloxacin, but they develop resistance to the other antibiotics that they've never seen because they're upregulating these multi-drug resistant efflux pumps. As I said, CSOM has high persister cell strains. And if the way that we test these in vitro is that we test MBEX, which is minimum biofilm eradication concentration. So how much does it take after we grow the biofilms of these bugs to kill them? So as you can see here, if we look at just the red and the orange, if we're now testing MBEC against these strains, it's not working. Another story is that iodine actually does potentially work in vitro, but not in vivo. So maybe the Australians got it right with treating the ears with iodine after all. But iodine does have limitations in terms of safety, toxicity, and pharmacokinetics into the middle ear. So it still isn't the answer. And so as you can see there, when we grow these bugs and measure the MBEC, it's probably around 3 to 750 when we know that the bottle concentration that's actually reaching into the area of where it needs to be is much below that. And this is the reason why we cannot control chronic suppurative otitis media with all our current therapeutics. So now I'm going to just change tack there and hopefully give some inspiration into where we may actually tackle these. So the first thing to realize is that, as we showed, the bacteria have low ATP, and this is a potential opportunity. So typically people think that bacterial cells that are sleepy are harder to kill. But if they have lower ATP and we were to leverage that, they actually might be easier to kill. And significantly, ATP is required in all. So able to that in some, we may be able to affect persister cells. And so we've been, another part of our lab has been trying to demonstrate this as a viable target for treating bacterial persister cells. The other thing to consider there is that the ATP also is vital in maintaining the respiratory train and metabolism and electron transport across the bacterial membrane, and also establishing a proton gradient, which is involved in repair of the bacterial outer membrane to damage. So it's required for life and it's required for repair. And what happens is when these cells go into persister cell states, they have to reduce their machinery. They shrink their ribosomes to do that, and they do that by sucking in the extracellular magnesium. And they shrink down the machinery to kind of what's minimally required, and they balance this out with a low ATP. And if you were to tip the scale in any direction, they really can die. But they kind of live on the edge is how you want to think about persister cells. And so one of the things that we understand in bacteria cell membranes, but particularly in persister cells, is that when they undergo damage in the outer membrane, there is a mechanism that happens there, and the damage to the outer membrane kind of leads to unfolded outer membrane proteins, or OMPs, and these are rescued through that mechanism there, and they kind of be refolded. If they're not refolded, they get inserted to the inner membrane, and you get mixing of contents between the periplasm and the inner membrane. The bacterial cell dies. So without this repair and rescue mechanism, the persister cells cannot function. So this is where I'm going to give a lot of credit to Laurent, who's a research scientist in the lab, who developed a strategy to go after these guys. So Laurent has developed a gold nanocluster approach to targeting bacterial persisters. We know that these are small, and we've played around with a couple of different sizes, which I'll go to in a little bit. But you can see there at the bottom right is the characterization of that, and they're probably in the order of 2 to 3 nanometers. And these are not nanoparticles, which tend to be larger. They tend to have a plasmon peak on a spectroscopy, which you can see on the top right. There's an absence of a peak, so we know that these are very small, and they're considered nanoclusters, and they kind of sit in this range here, somewhere between the size of a glucose molecule and an antibody, in the size of, we're playing around in the realm of 4 nanometers. And we don't want to lump nanoparticles and nanoclusters and all the approaches together. And that's just giving you some different approaches that have been done on the bottom left there. You can see that the size ranges that people typically tend to play with are around about 100 nanometers in the nanoparticle range. And nanoparticles tend to form a protein corona and in the blood and have different interactions, whereas nanoclusters do not. They have very different ways of acting. So when we started to play around with this, we actually found a couple of interesting things. So this was an experiment where we measured the OMP, remember the outer membrane protein folding. And remember, if it becomes folded or unfolded, it can have different consequences. So using this blot here, you can see using ATP on different increasing concentrations of ATP, you can see that the OMP exists as a monomer. And then if we were to use a gold nanocluster functionalized with something else, which is another nanocluster approach, you can see it still exists as a monomer. But strangely, when we were to functionalize it with ATP, gold nanoclusters functionalized with ATP, something started to change. We see that there was folding or transition between a monomer and a trimer as you increase the concentration. So we were interested in this observation where potentially the gold nanocluster could affect the folding of the outer membrane proteins. And what we hypothesized is that there was some level of damage that could be incurred to the outer membrane, and that if we prevented unfolding of the outer membrane proteins, we could potentially lead to damage of the inner membrane, like I alluded to before. One thing is that if you can see here, I think I've just covered that up there. On the bottom left there, you can see there's CLIP protease that rescues this. So we know that there are knockout bacteria that we can knock out CLIP-XP. So what we've done here in this experiment is if we've tested PA14, which is a different strain of Pseudomonas, and on the left there we have the wild type, and on the right we have the CLIP-X knockout. And you can see there, if we increase the concentration from blue to purple there on the left, and we're going from 0 to 2.3, well, if we knock out ClipX2, so we're knocking out the ability to rescue the unfolded membrane proteins there, you can see that it sensitizes to the gold nanoclusters in going from 2.2 to 0.2. And so what that is showing us is that that potentially confirms the mechanism of action here, is that may be true, that it that affects the folding of the outer membrane protein. What we also did is that there's an experiment where you can do where you can give propydium iodide, and it fluoresces if there's injury to the inner membrane, because it's able to go inside the cell. And so looking at Pseudomonas, E. coli, and Klebsiella MDRs there, we can see there it goes from the blue is the control, the untreated, and the red is the colistin, which is known to lyse cells as the positive control. You can see there that we're able to increase the excitation, which is a surrogate for the inner membrane integrity of the cell there. So we know that we are very likely disrupting the inner membrane in this process. But very interestingly, as you can see on the left there, we're able to kill multi-drug resistant Pseudomonas E. coli and Klebsiella, and we're able to do that and sterilize them using our nanoclusters. And so, interestingly, if you were to pass those sterile where we've killed the bacteria, and then we shake them up and take the supernatant out of there, and we have a couple of controls there. We have the fluoroquinolone antibiotic and the colistin antibiotic there, and we measure the protein concentration in the supernatant. We can see that we can measure that, but you can't measure that in the gold nanocluster exposed ones. So what is that is telling us is that for some reason we're able to cause cell death or inner membrane permeability without causing outer membrane permeability and causing cell lysis. There's some data here that I'm not showing that actually confirms the outer membrane integrity. But we look at these under the electron microscope. On the bottom left, you can see what Pseudomonas typically look like. In the middle there, you can see what they look like after exposure to ATP, the gold nanocluster with ATP. They look the same. And on the right, you can see what happens at super concentrations of afloxacin. And so, interestingly, these cells seem to be dying without lysing. If you look even closer at these, there seems to be some blebbing that occurs on the vesicles of the outer membranes of these bacteria. And what we hypothesize is that maybe these are vesicles of misfolded proteins, which can be shown in with other models as potentially existing. So obviously we need to do more work here to show this, but it's kind of our hypothesis is that we potentially are acting on these folding of outer membrane proteins. Interestingly, this is the first time that we've been able to show that persistent cells are easier to kill than exponentially or metabolically active cells. So they're susceptible at lower concentrations. You have to drive the concentration higher up to kill these exponentially dividing cells as opposed to these persistent cells. And if you passage them over time, on the top one you can see there, if you give it without that, they don't develop resistance after serial passage. But also you can prevent resistance from happening if you co-administer it with antibiotics as well. And also prevents cross-resistance, which just for interest of time, I'm not showing you the data. Interestingly as well, it likely reduces the virulence. So you can see there, piocinin, which is a virulence factor of Pseudomonas, goes away when we treat it with the gold nanoclusters. And if we go looking back at the multi-drug resistant bacteria, we seem to be able to kill most of the things that we're able to test so far with Pseudomonas, Staph, E. coli, and Klebsiella extensively drug resistant. And XDR means that there's no currently available antibiotic which is able to kill these bacteria. So now we're going back to our clinical isolate bank, and thanks to our collaborators in India and Egypt. And you can see here, looking at the patients, these are the blue is the vehicle, and the gold nanocluster is in the red. And you can see the massive log reductions and in most cases sterilization of all patients. We did also compare this to with ciprofloxacin, which is not graphed here, showing that they were also not susceptible to ciprofloxacin in most cases. Some of them had unrecordable MICs as well. Again, looking at India and the first 3 rows on the right and Egypt on the bottom, able to sterilize almost all of the samples that we have. And we go back to the animal model that I showed you earlier. And we have we've also tested this against fluoroquinolines as well in another graph that I'm not showing you here, showing that we're unable to treat the infections or sterilize the infections with fluoroquinolines. But we're able to get dramatic reduction and in most cases sterilization in vivo. And so that was really exciting to show that in vitro we're able to convert these to in vivo potentially cure of chronic suppurative otitis media, at least in our animal model. We're a very translational, translationally focused lab. These are ABRs. We do have DPOEs, which I'm not showing you for the interest of time. The negative control on the left. On the right is a positive control, which we do with iodine. We can see an elevation of the threshold in green. On the bottom left, you can see that at 10x the dose that we're going in vivo, there is no elevation, but we do see an elevation in thresholds at 100x. Very unlikely that we're going to have to go 100x in our patients, but this at least gives us a profile of safety. When we looked at where this threshold elevation potentially came from, we looked at the inner ear, and the inner ears had preserved hair cells. In fact, very little gold on the right actually gets into the cochlea. And where this threshold elevation comes from is at super concentrations at 100x, there seems to be a fibrous reaction in the tympanic membrane. When you look at these, they look really thickened. And so it seems to stimulate an almost an overreaction in the fibroblast repair mechanism of the drum. So, not sure why that happens. It's an area for further investigation, but at least this is telling us where the toxicity may lie, and hopefully it's not in the inner ear. Very little gets into the blood. So we looked at pharmacokinetics into the blood. A small spike happens within 24 hours, less than 0.3%. And just getting towards the end there, a really interesting phenomenon that we observed is you can see on there, if we play around with different sizes, 0.8, 1.2, 4, they even look different when we synthesize them differently. But some, if we play with 0.8, it actually doesn't control the infection. If we play with 1.2 and 4, we can eradicate it. And so we just looked at, big versus small in terms of size, 1.2 versus 4. And you can see there that in at least in this test, only the small had the effect, whereas the big didn't. So even a minute change in size seemed to affect the properties of the biophysical properties of these in the way they interact, of which we're still trying to comprehend and understand. But at least a very interesting scientific phenomenon. And just showing you again the small on the top right, big in the middle, and the controls down the bottom right. You can see that they seem to have different plays with that OMP assay that we did before. The big seems to be changing the folding in the middle regions, whereas the small didn't. So this is just confirming that the sizes look different and behave different. And so I know I ran through a lot of material there, and I know there's a lot to take home. So I'm going to summarize it very, very easily for everyone. So I just want the take-home message for everyone is to understand this concept of persister cells. It's the reason why chronic suppurative otitis media doesn't get better. It's the reason why it relapses. It's the reason why it's hard to treat. And so whenever we think of chronic otitis media or chronic bacterial infections of any kind, we need to be thinking of persister cells. And on that note, we have a ton of people to thank, both across Stanford and in Pittsburgh, people that have worked in our lab in the past and in the present, a huge group of people to thank in terms of funding, the Royal National Institute for the Deaf in the UK, the NIH across a number of different institutes, internal funding both in Stanford and Pittsburgh. And I wanted to thank everyone for letting me talk about something that I'm very passionate about. And of course, happy to answer questions and follow up with anyone. Thank you very much.
All right, great. Thank you so much. It's very great, interesting talk. If anybody has questions, feel free to go ahead and unmute now and ask. I didn't see any in the chat, so I'll just give you a minute to do that. Well, while people are thinking and taking all that in, I guess from an audiology, like clinical perspective, we see kids with ear infections all the time. A lot of them get treated and never come back, or they get PE tubes. Like, at what point does it switch from just a normal ear infection to something more serious where these persister cells start showing up? Like, is it just simply time, or are there certain things that go into that?
Yeah, I think, and what we understood about that is when we started to set up the model. So that model took us about 18 months to 2 years to get right. And you'll see a lot of models of otitis media, and they're mainly acute models, and they'll provide an inoculate into the middle ear, and there's interaction between the host, which is usually the immune system, and the bacteria, and the bacteria will either win, and they'll cause a labyrinthitis or meningitis or sepsis and things like that, or the host will win and they'll clear the infection. If the insult at the beginning is too much, you're up the host is gathering the immune system and saying, "Yeah, we got to get rid of these guys, else we're going to be overwhelmed." So the way that we inoculate is right in the middle. So we do it very consistently. Now we've repeated this model 30 times in different settings, but when we gave it at first, we would give an inoculate that would lead to labyrinthitis, meningitis, and things like that. So it would be too much. And others, we would give an inoculate and it would last maybe a few days or a week and it would be cleared very easily. What we now do is cycle through and inoculate with persister cells, and we give we grow the bacteria, we treat them with ciprofloxacin, we get the results, and then we put that into the middle ear. So we're sort of, these are grumbling along, they kind of wake up slowly. They don't really set off the alarm bells of the immune system, and they don't clear it. So I think that's what we're seeing in the human setting. Is in most, as we know, the vast majority of otitis media that we see is acute in the acute setting. And mostly this is an interaction where you have an immune competent host which is clearing it, and sometimes you need a little bit of over-the-top help with antibiotics that help it clear a little bit faster. But there is some point in time where if that thing is a little bit subclinical, where it can set in, and clearly the bacteriology is at play here. So we know that acute otitis media is a very different bacteriology to chronic suppurative otitis media, and you'll see that most of those are skin bugs that we're seeing there, and they're probably getting in through infected water or infected ears, all that sort of things. And so the bacteria are probably different. They behave different. They're not setting off the alarm bells, and they're probably designed to have not quite a symbiosis, but to fly under the radar a little bit. Some sort of work that we've also done, and that we'd love to explore more, but we had limited opportunity, is that we've also shown that these Pseudomonas in particular that we've studied tend to cause the immune system. They come in, they kind of bring in a neutrophil response early. They bring in an immature neutrophil response, and these immature neutrophils are more likely to die when they put out their neutrophil extracellular traps instead of to survive. And when they die, they release a lot of their cellular DNA, and these often become the building blocks of the biofilms. So it's almost like the bacteria have evolved to call in the host to cause the immaturity of the host immune response and cause them to build into the system. So, that's a long way of answering your question of getting there, but it's at some level an interaction between the host and the bacteria. I think there was a comment from Robert about, can I comment about why I didn't sterilize the specimens from all the patients? Yeah, that's a good question. So in our test, we did this in vitro, and we did one treatment. And when you look at what the FDA defines as antimicrobial activity, they look for 3 to 4 log reduction. So we passed the bar from that, and we're hoping that maybe there's issues of pharmacokinetics where it's not penetrating in there. I mean, when we did it in vitro, we're pouring into plates. When we're doing it in vivo, we're literally just pouring it into the ear canal. And so maybe it's not getting through all the areas that it needs to be, and these are reactivating in somewhat, and maybe these are able to clear with multiple treatments. So we're hoping that at least that with more than one treatment, we might be able to sterilize or get close to that. Yes, a question from Hella about, thanks Hella, giving the evidence suggesting a genetic susceptibility to otitis media. I'm curious to know if this is something you would explore further. I would, I would love to. I would love to work with you, Hella, to explore this further, because I think that you're a genetics expert and I am not. But certainly, if you look at this from two different angles, you look at the populations that are affected. Clearly, this goes beyond socioeconomic conditions. You know, why are the Australian Indigenous communities, other Indigenous communities, affected more than others? All right, even when they, if they were, you know, to grow up in the cities as opposed to rural, they are still affected more. And so there's clearly a genetic component which we know about in the clinical setting. From a actual discovery science point of view, I think I have too many hypotheses, and I need some help to know where to start looking at that. And so I'd appreciate advice and collaborations from people like you, Hella.
Thank you.
Hi, Peter. I also have a question. So some of the drugs that you mentioned, like Cipro, they also have an effect on the ciliary beating and the mucociliary clearance from the middle ear. And also even your solution to use ATP also has an effect on the ciliary beating frequency, and definitely going to affect the clearance. Have you looked at it from that perspective and what's going to happen from
Yeah,
that?
not at all. We didn't consider that at all. So we haven't looked at that. That'd be interesting. As you know, what role does eustachian tube dysfunction have in this? You know, clearly otitis media and eustachian dysfunction are linked. How much is needed to be effective? When at least in these mice and in most of the human patient, it's probably overwhelmed to the point that it's completely dysfunctional and it's blocked. But how much of that do you need to open up and clear? So yeah, no, we didn't look at that. It'd be really interesting to know and know if we are affecting that positively or negatively, and how is that maybe leading to the residual ones that we're able to clear.
Yeah, that would be interesting. Also, like it has an effect, definitely, like the ciliary beat frequency, also connected with immunity, that kind of you touch, based upon a little bit, and the release of peptides that also they might have a role. But very interesting.
Yeah, I think there's other interesting mechanisms that can happen. Obviously, we're going at the primary, but there's very other secondary mechanisms. And I think the thing that we're trying to understand is, you know, a lot of things work out in plates, but they don't work out in vivo. So we've got to understand pharmacokinetics, drug delivery in the middle ear. We've got to know where these things are hiding and how to get to them. And it's not just sitting on the surface, which is really what the fluoroquinolones are treating. It's, you know, within the cells. There's evidence that persister cells are living inside macrophages and neutrophils within the cells or within epithelia, and that maybe these are reactivating upon cell death and things like that. So I think there's a whole bunch of other mechanisms that we need to explore.
Thank you so much.
Thank you. Anybody else have questions? I don't see anything else in the chat. No. All right. Well, thank you everyone for joining, and thank you, Peter, for a wonderful talk.
No, my pleasure. And as you can tell, I'm super excited about this work. So if anyone ever wants to contact me, this is an opportunity to call for collaborators and people to talk to about this. So please reach out. And thank you all.
All right. Thank you.
See everyone.